Okay, I think we've just about reached critical mass, so I think we're going to go ahead and get things started. Good afternoon, everyone. My name is Ryan Leung, Director of Oncology Product Development here at PathAI. Really excited to welcome you all today to today's webinar with Ilan Wapinski, PathAI's Vice President of Translational Research and Algorithm Products, to learn more about the promising role of the tumor microenvironment and how artificial intelligence-powered models can unlock new capabilities and insights for drug discovery and development. Over the course of the presentation, we welcome you to share questions through the Zoom Q&A function. But please note, we will be waiting until the end of the presentation before answering them. We'll be keeping track of any questions as they come in, but we'll be waiting until the end of the presentation before addressing them. Also, please note that this session is being recorded, so please reach out if that would be of interest for any folks that are not able to attend or may have to leave early. And with that, I'll hand off to Ilan.
Thank you, Ryan. Thank you so much. Very happy to be here to be presenting to this fine audience. The title of this presentation is Unlocking Novel Insights in Oncological Drug Discovery, Drug Development with the Tumor Microenvironment. My name again is Ilan Wapinski. I'm the VP of Translational Research and Algorithm Products here at PathAI. Before I get any further, I just want to highlight that this webinar is solely for educational purposes, and the products and services discussed are for research use only, not intended for diagnosis or treatment. All right, so I'll start with just a high-level agenda and what we're going to talk about, and then I'll introduce the themes and the topics that we're going to go into some case studies. So first, I'm just going to talk about the histopathology image analysis of the tumor microenvironment, or TME. I'll use those two terms kind of interchangeably throughout the discussion here. We'll talk about how we analyze clinically relevant spatial relationships within the features of the TME using human interpretable features. I'll discuss a few case studies, such as predicting disease progression in ovarian cancer using some nuclear features that we extract from the TME, and also predicting structures such as tertiary lymphoid structure signatures or c-Met expression patterns using H&E images of the tumor microenvironment. Finally, we'll have some Q&A that will be moderated by Ryan, my colleague here. So thank you very much for attending. I hope this is of interest. I do want to also start by introducing PathAI to some audience members. For those of you who are not familiar with PathAI, we're a digital pathology and laboratory company. It seeks to improve patient outcomes through AI-powered pathology. And so, as most of you know, pathology plays a very critical role for many serious diseases, including almost every solid cancer. It's important for drug development and clinical trials, as well as every patient being diagnosed and treated for cancer in the world. We think there's a lot of information stored in each of these whole slide images of tissue specimens such as this one that are routinely collected at the onset and sometimes even further on throughout the patient journey. Historically, this information has not really been fully captured or appreciated or unlocked to better identify which patients will benefit most from which biomarker-directed therapies. So a huge mission here at PathAI is to unlock this potential and develop new approaches to improve accuracy in diagnosis and aid with clinical development. So the cases that I'll describe today are focused mostly on exploratory research, but we always keep an eye towards the future, where we think every patient in the world will have access to this technology that should be available to enable optimal treatment for each individual patient, so they can deliver on really the promise of precision medicine. Right? So with that, I will kind of introduce, this is kind of an H&E image that we'll be spending a little bit of time on today, and the insights that I'll talk about really are in the early stages of where we think this whole technology and the platform is going. So I'll be describing some of that and how it enables us to apply our AI-powered approach towards unlocking all the information about the tumor microenvironment that's contained within images such as this one. At the end of the day, and kind of importantly, we think cancer is really defined as a disease of cellular spatial location. It's not really by genetics or RNA-seq or protein expression. Cancer really is defined by malignant cells invading into normal tissue. And that's a pattern that we can identify by eye, such as here in this image. But that identification, that process has its limitations. So there's lack of reproducibility in identifying these patterns that we know are important. And there's some patterns that are hard to discern or even kind of hidden or undiscoverable just through the naked eye alone. So in this H&E image of breast cancer, we see kind of at low power. You already see kind of some of the structures and morphologies and different features that are available for these types of analyses. So you see, again, I'm not a pathologist, but I'll say like you can see areas of tumor, areas of stroma, areas of ductal carcinoma in situ over here. And the entire microenvironment, the necrosis, the epistromal interface, all of that, it's really available for us to capture from these H&E images. For pathologists, they know and they realize through their jobs, right, that this is untapped and unmined information. But to someone like myself or researchers or business leaders who aren't really pathologists, it might not always be obvious that there's so much information in an image that's routinely collected like this. So here at PathAI, so we've trained models to recognize a lot of these patterns that we know are important and can be applied at a very high scale and accuracy. So in this case, we have an algorithm that we've trained here that can identify the tissue and cell types in the TME to enable accurate and scalable and objective identification of these clinically significant features. So you could see various tissue types here, the tumor epithelium and the stroma, like I was describing before, but you could also see the DCIS areas that I was highlighting. And the AI model can now be overlaid onto this H&E image to give a sense of what the model is thinking, what the machine is predicting for each and every pixel, and can be viewed on our AI site, Digital Pathology Viewer. So this is kind of like a high level, right? The first thing that we look at is where's there tissue? What kinds of tissue are there? There could be some tissue types that are a little bit more maybe esoteric, a little more sophisticated, things like tertiary lymphoid structures. So here I'm highlighting that areas of the tissue that we predicted to have mature tertiary lymphoid structures even with germinal centers or immature TLSs. And we think that there's a lot of power in being able to capture each and every one of these features, count them, identify where they are located across the tissue, where they are with respect to one another. And of course, we can overlay this along with the heat map that I showed you before, and you could kind of start to put these things together and see areas, for instance, of inflamed stroma that are indicative of maybe nascent lymphoid structures. Right? So we'll spend a little bit more time on images like this in a few minutes, but I first kind of want to introduce these questions of at a high level, what are some of the features that we're trying to capture within the tumor microenvironment through AI-powered pathology? So our approach is to quantitatively characterize the tumor microenvironment through these algorithms, through this technology, to unlock new insights into disease biology and inform biomarker strategies by identifying properties of the TME, such as the immune landscape or vascular network, to help determine patient prognosis and response to therapy. And here are just a small sampling of some recent publications that highlight the importance of some of these features, such as stromal subtypes or tumor-infiltrating lymphocytes that has gotten a lot of attention recently, that we can really now identify systematically and at scale through our technology. So let's go back to that sample that we looked at before. I zoomed in a little bit further, and here I'm highlighting in red what the area of the tumor parenchyma are, but what I'm not highlighting with our overlays are the area of the tumor stroma, and you could see right away that it's not all stroma looks the same, right? That you could see there's quite a bit of heterogeneity and differences between areas of a lot of cellularity or a lot of collagen, the orientation and structure so that are present within the stroma are also perhaps very indicative. So one of the approaches that we've taken here is to train models that capture some of this heterogeneity and to try to understand what is biologically or clinically relevant. So for instance, here we're showing the overlays of our 5-class stromal subtype model that can characterize areas of fibroblastic stroma or inflamed stroma, or even mature, immature stroma, et cetera, that can be overlaid on top of areas where we think there are stroma. So this just kind of highlights some of the layers of complexity, kind of really like layers of an onion that we're peeling back, and we're kind of understanding what is present within each and every H&E image, what is present in each tumor sample that we can capture, that is really indicative of the disease biology for this patient, and that could also help inform what is the best course of therapy for this patient, perhaps, or what are some of the treatments that one could design and develop in order to address these different aspects of this disease. So this is kind of the nascent period of this type of approach. There will be a lot more to come on how we characterize, featurize, and analyze features like this of this stroma. So we really feel like we're just scratching the surface in this case, right? And so I'm going to zoom in a little bit more, right? So what about cells? So far, everything I've talked about is all about the tissue regions, stroma, necrosis, tubular epithelium. Of course, we can identify each and every cell on this tissue. So I zoomed in, and I'm showing you here where there's cancer cells, where there's lymphocytes, plasma cells, macrophages, and fibroblasts. So these are some of the important cell types that one can look at. Of course, now you can systematically count and quantify all the tumor-infiltrating lymphocytes in samples such as this, and then use these features once again in downstream analyses. I'm going to zoom in just a little bit more because not only can we quantify each and every cell, but we could actually even characterize their shape. We could see which ones are rounder, which ones are longer, which ones have a lot of eccentricity in their morphology. What is the heterogeneity of the chromogen within each and every nucleus? So again, these are all things that one could capture as accurately as possible, as accurate as the technology allows us to do, in each and every whole slide image that's routinely collected throughout through the kind of the patient diagnosis journey. And we think all this information is very important, and it really kind of puts this holistic picture and informs some of the very important hallmarks of cancer and disease characterization that one can use. So in summary, we think that this is important because we really now have the tools and the ability to analyze each and every pixel, each and every cell, corner and cranny of the whole slide image. And it opens up our ability to investigate all these different histological activities and patterns that underlie, in some cases, the molecular or other sort of genomic features of the patients and their clinical manifestations as well. Okay, so what I've shown you already is that AI can augment and enhance what we could see with more clarity, granularity, reduce inter-observer variability, improve sensitivity. We could standardize the assessment at an unprecedented scale. But I'm also going to show in some of these case studies how this approach can really be powerful in its ability to interrogate the different spatial orientations of these different features and arrangements within with respect to one another, and reveal some things that the human eye or brain can't really see or compute. These are very sophisticated AI techniques that we've adopted here that could be used to predict patient outcomes or identify novel signatures that correlate to prognosis or diagnosis, or that can capture different aspects of heterogeneity across this whole slide images with new accuracy and speed. So we'll spend a little bit of time talking about graph neural networks and how we use them to predict certain signatures. Before I dive into that, I'm going to talk a little bit more in simple terms about our kind of human interpretable features. So human interpretable features are this way that we think about all the kind of a quantitative summarization of the features in each sample that we analyze. So it kind of allows a kind of a simplistic way of capturing what's present in each sample. So this was I guess a term, HIFs, is what we're using, that we first described in a publication that came out last year in Nature Communications, and it demonstrated how we use these human interpretable features to associate them with different genetic markers or expression patterns. I think of HIFs as kind of a standardized way of summarizing each slide in a quantitative histopathology kind of feature table that can be analyzed similarly to how one might analyze RNA-seq or other genomic data that are now quite routinely available and most people are very familiar with. One can think of them as kind of like another orthogonal data set. So going into some of these case studies, I'm going to talk about how we apply nuclear features to predict disease progression or outcomes in ovarian cancer. This is some work that was recently shared at ESMO earlier this month. We're also going to talk about how we predict signatures of tertiary lymphoid structures using GNNs, and that was work that was presented at AACR a few months back. And just last month, we also used GNNs and HIFs to predict c-Met status, c-Met overexpression status in lung cancer at the World Lung Congress in Vienna. So excited to dive into some of these samples. All right, so the first case study here. So we create AI models that characterize a tumor microenvironment and the nuclear morphology in high-grade serous ovarian cancer. This was a collaboration with researchers at Cleveland Clinic, where we tried to use this approach to characterize the TME and predict features of nuclear shape and morphology that were then associated with disease progression. So first, just to describe at a high level, of course, how we develop these TME models. So we collect very many thousands of pathologist annotations. We train models, giving the models examples of what different substances of interest, different objects look like. We tell a model, this is what stroma looks like, this is what a lymphocyte looks like, this is what a cancer cell looks like. We do this many, many times, and the model becomes increasingly more accurate over time. And then we apply these models, as you could see through these overlays and the overlays that I showed previously, to this data set of ovarian cancer. Similarly, we did a similar approach for identifying each and every nucleus and characterizing which pixels belong to a nucleus, whether that nucleus is a cancer cell or an immune cell, and then extract these features that we then associate with the state of the patients, right? So one important feature here is that we validate these models. We know that they're accurate or as best as we can by comparing them to that of a consensus of pathologists, and that's what the data that I'm showing you here. So this is some results that I'm sharing on TCGA data. So one thing that we found kind of interesting even before we looked at our features is this characteristic of cancer, of course, is aneuploidy. This is often present in lots of patient samples. And it turns out, not surprisingly, that in this data set, aneuploidy gains is actually quite predictive of the patient outcomes. This is again, just straight from the TCGA data set. These are outcomes that were derived from that data set. And we're also able to then associate some of the features of the nuclei, the cancer nuclei, with this aneuploidy status in this data set. So in this case, we looked at the nuclear minor axis length. So what does that mean? So we look at each and every nucleus, we look at what the major axis is, what the minor axis is, and we then quantify for each and every cell what that length is and what's the variability across the sample. So here's, for instance, an image of a patient where there's a lot of variability on this minor axis length, and we could see that this feature is associated with this aneuploidy status in this data set. So this feature, not only that, is also associated, not surprisingly, but importantly here, it's this nuclear feature that we're deriving direct from the H&E image without access directly to any genomic information is also attributable to disease progression in this data set. So this was, like I said, presented earlier this month at ESMO, and we are happy to share some of those results at a future time. Feel free to contact us. For that, I'll go to the next example, which is predicting tertiary lymphoid structure signatures, again, from H&E images. So some of these features are of great interest. They've been associated with disease progression. They've been associated with response to certain therapies in immuno-oncology and other settings as well. And here, like as I showed before, we can quantify the presence or absence and the degree of presence of these tertiary lymphoid structures across these samples. So what we did here is use the features of the tumor microenvironment to train a graph neural network to predict the gene expression signatures of some chemokines that are associated with these TLSs. So let me spend a second describing what a GNN is, a graph neural network. So this is kind of a new paradigm in machine learning. And what we do is we take the outputs of our heat map. So we have trained the models to predict where the cell types are. Now you imagine they're arranged on some sort of topology, right? They're across some sort of landscape on the tissue. And you can use that topology to describe a graph. So nodes on that topology that are near each other can get adjacency, edges between them. And that graph can be used in itself as inputs to train a model. So what's important about that is it's not only telling you what is present within that tumor, but it's telling you the spatial localization with respect to other features of that tumor. And so one nice way of thinking about this, of course, is kind of if the human interpretable feature table, the HIF table, is kind of like that smoothie, the GNN is kind of like the fruit salad. It's telling you what's next to what. So you're not just blending all of the cell types together, you're not blending all of them as one kind of vector or feature, you're actually looking at how they're related to one another across space on the tissue. And that's notably very important, as we said before, and we find this paradigm quite important for predicting outcomes. So that's what a GNN is. We applied this approach across different tumor types, again, using TCGA data here, to train models to predict the expression patterns of these 12 chemokine gene expression signatures, and then predict that on a held out data set. So once we train a model using GNNs to predict what's the expected expression level of these different gene signatures, we deploy that on new data and infer what we would imagine, again, without knowing anything about the actual molecular status. We're imagining we don't have the gene expression directly from those samples. We just have the imputed expression signatures of those samples, and only cluster different patients based on this imputed expression pattern. So this is kind of the predicted signatures 1 through 12, and then we were able to kind of cluster the two patient populations into one cluster, another cluster, based on these imputed signatures. And that worked out quite well. And what was actually really remarkable was that if you look at the prognosis, the kind of the disease state of these different patient pools, we see that one has a much better prognosis than the other, just based on what the GNNs are predicting to be present. We could also interrogate the GNNs and ask it, like, what is it that you're picking up on? What are the regions on the tissue that you think are interesting, that are relevant for predicting these chemokine signatures? And because we have those human interpretable models and we can look at what the model is thinking on each and every pixel, we could overlay the tertiary lymphoid structure predictions onto the GNN predictions and see that by and large, there's a pretty big overlap between what the model thinks are relevant for predicting these chemokine signatures, but there's also some areas that are actually kind of exceptions that are relevant for, or that are related to these chemokine signature predictions that are not specifically on where each and every imputed TLS is. We thought that was also very interesting and important. All right, so for the last sort of vignette that we're going to introduce, I'm going to talk about how we apply this approach once again to predict the CMET status in a group of non-small cell lung carcinoma patients. So this is kind of a pretty known marker. It's in CMET dysregulation. It's known as a proto-oncogene. There's different ways of measuring it through IHC or through gene expression or other kind of molecular approaches. And here what we're using is, again, the H&E image, the tumor microenvironment, and how we characterize it to predict the CMET status. And this was presented last month at World Lung. And what we're able to see is, at least in this data set, we're able to predict on a held out data set that the model is quite sensitive to predicting the CMET status, the expression status on data that the model was not trained on. It's quite sensitive here, as you can see. And then again, we could interrogate what is it that the model is seeing? What are the features in the tumor microenvironment that are most relevant? And we see that, again, the density of lymphocytes within the cancer epithelium, these are one of the features that are most predictive of the CMET status in these patients. All right, so that was kind of a summary, 3 little stories. We have many more that we could share, but we wanted to be mindful of time. We also have a few stories that are coming up in some future venues. We have one coming up later in October that kind of describes a tumor immune microenvironment atlas that can across many thousands of cases. That will be presented at Path Visions. And then at SITC, we have 5 abstracts. A few of these are on the H&E based analyses. Some of these are on immunofluorescence or IHC. So everything that I described so far here is on H&E based analyses, but of course we could apply this approach and apply a lot of these same learnings and the same technological platform to assess IHC or other kinds of methods, right? Okay, and finally, I will also just highlight, we are eager to kind of open up ourselves to kind of more opportunities to partner with us and to advance the field using this technology and applying it to other disease areas or data sets. We could deploy our algorithms kind of at scale. We have quite a few in different disease areas: breast, colorectal, gastric, melanoma, et cetera. And we're also, of course, able to kind of partner with partners such as yourself. We have access to large data sets through our partnerships with Datavant and Cleveland Clinic that can help us train models that perhaps we don't have already kind of in our arsenal, or that could be relevant for answering very specific questions that different partners might have. And finally, like if you're interested in kind of using these GNN approaches for understanding spatial phenotypes, we can scope out kind of custom translational research projects using our translational research liaisons to be dedicated to kind of scoping the right project to work across both internal project teams. We have many hundreds of engineers here, or hundreds of engineers, many dozens of engineers, and to work with your groups on clinically relevant studies. All right, so that will end us at about plenty of time to share. Any questions or other concerns?
Awesome. Thank you, Ilan. So I'll just welcome folks to continue adding questions in through the Q&A function, but in the meantime, can start to take off those that were brought up during the course of the presentation. And so to start things off, Ilan, when developing your TME algorithms, how do you determine what features of the tumor microenvironment to capture? And then sort of a follow-on to that is, does that vary based on the indication or the use case in your view?
Yeah, so I'll answer that. It varies quite a bit. So how do we determine? We have a team of excellent pathologists here at PathAI. So we have kind of 2 groups of pathologists that we work with. One is our in-house pathologists that are a very key part of all of our model development efforts, from scoping to making sure all the way through development, and making sure that we're capturing all the most critical features in that data set, and that can inform the most about disease biology. So an HCC data set might have features that are very different from a breast cancer data set or bladder cancer data set. So there is a lot of kind of domain specific information that we want to capture. We want to be as both clinically and biologically relevant as possible. How do we decide? So based on, we try to take an unbiased approach. We want to capture each and every cell type that we can. Some cell types are more present in the data sets than others. So we'll have better predictive accuracy on those data sets that are more present or that have a more consistent presentation than others. But we think that nonetheless, even in the areas where there are fewer of those data sets, we can still capture enough information there that is relevant for downstream analyses. I think that captures the question that was posed, but let me know if there's anything there that I missed.
I think that's great. And if there's any clarification that someone who asked that question would like to have, please just feel free to add in a follow-up.
Okay.
Continuing along, can you speak to the biological relevance of the various stromal subtypes? And I think this is sort of related also in terms of the definitions as you were describing it for us to define these substances and how we identify those that we think are going to be biologically or clinically relevant.
Yeah, so obviously, like the most important aspect of each of these data sets is kind of to test it on the data set that's being studied, right? So the role here of these subtypes is to kind of characterize the subtypes. We understand based on the literature that there are kind of associations with these different subtypes with disease severity, prognosis, in some cases with the ability to, for certain treatments to kind of access the tumor. So we designed these models to be as kind of comprehensive as possible based on what we were able to garner from the literature. Different subtypes will be have different relevance in different tumor types. I think that's kind of goes with the previous answer. The subtypes in pancreatic cancer or renal cell carcinoma are going to be very different, have different relevance to the patients than breast cancer or lung cancer. The models hopefully are going to capture as much as possible and be as accurate as possible, so that when you're analyzing those features with those data sets, the outcomes data or the response, the treatments data, you can actually discover for yourself which are the relevant biomarkers. So that's the stromal subtypes, yeah.
And just to add briefly, I think, Ilan, you'd sort of already hinted at this, but this is not a static process either. Over time, as new substances are defined and as we continue to unlock additional insights, I think the beauty of the approach is that, of course, these models can continue to evolve over time. And again, be taking in the latest in biology and scientific research as it uncovers.
Yeah. I think one important thing here is we try to be as unbiased as possible, right? So we could be very hypothesis driven, and that's a really important thing to do, right? We have a hypothesis that, say, mature stroma is going to be associated with response to therapy in this indication or that indication. And so let's design a model that really captures mature stroma. That's very important, that's useful to do. In our case, we want to just bring as much information to the analysis as possible and let the data kind of take us where it will.
Great. Another question here. Any thoughts on using IHC rather than H&E to refine cell identification and prediction of genetic mutations? I think even more broadly, I guess, is where do you see opportunity for us to be leveraging multiple biological modalities potentially to train algorithms, or where some modalities may be more appropriate for certain tasks than others?
Yeah, so we've done this, and we are very eager to apply this approach. So in the case of CMET or other indications where there's kind of there's an IHC kind of companion sample with the H&E, we definitely want to leverage as much information as possible. The models and the approach to training the models is very similar on IHC or H&E. Not every IHC is as robust or as standardized as the H&Es. H&Es are ubiquitous, right? Every sample has an H&E. It's kind of the gold standard. Not every sample is going to get a CMET IHC or a PDL1 or a CD8. In fact, in many cases, they're not going to get many of those things. So the focus here was really on H&E, but one thing that I also think is important to note from this question is, can we leverage IHC to help us identify what's relevant within the H&E image? And I think absolutely, right? So we have IHC markers for different cell types, and that can help us identify where there's areas of immune infiltrate, or where there's markers of say, that are macrophage specific that can be helpful for identifying where the macrophages are on the H&E image. So we leverage it both ways, and these kind of combined biomarkers are definitely something that we've been exploring.
Great. With everything that you'd sort of described with this ability to calculate a very large number of human interpretable features, how do you think about stratification when there are multiple variables that are popping up? Are there processes perhaps for understanding potentially confounding signal?
Yeah, excellent question. Not just confounding signal, but oftentimes, we have to be these different features can be correlated with one another and highly correlated. So you want to be careful in how you interpret them. You want to so we have a large team of data scientists who have a good background in understanding how to correct for a lot of these confounding signatures, or also kind of just do the proper corrections for the multiple hypotheses that we're testing each time. There's different approaches for doing this. So the first thing is that we try to pick kind of representative features, so that we're not looking at hundreds or thousands of features all at once. We kind of can cluster them together. We know that these features kind of correlate with one another, they're associated with one another. So we would pick features that are more robust or more consistent across the data set. They tend to be features that are a little bit more simple to define. And that's definitely a concern. I mean, it's something that we are aware of, and not just we are careful not to kind of fall into this trap. And ultimately, whatever signatures that we find, we're going to be testing on some held out data set. So we know that the truth validation is whether this signature is robust to new data. So that's something that we're also very mindful of. We don't want to just kind of overfit because we have such a high dimensional data set. We can predict almost any signature.
Awesome. And so you mentioned that PathAI leverages a large network of pathologists, both internal and external, to train its algorithms. Can you speak to how PathAI verifies and validates the performance of those models?
Yeah, I think I have a slide on this, but yeah, that's an excellent question. Whoever asked that. So yeah, so how do we know that the model is accurate? So there's a few ways to validate. And first of all, like what's really nice about pathology, unlike say molecular predictions, how do you know that this gene is overexpressed in this patient or not, right? You could kind of look at gene expression and maybe do some subsequent validation on other samples from the same patient. But with pathology, you can see what the model is seeing, right? You could see like it thinks this pixel is a cancer cell, or it thinks this pixel is a lymphocyte. And then you can kind of compare that to trained pathologists that are in-house to kind of QC the models. You could also use a kind of more systematic approach and try to validate using, actually, I'll jump to this extra slide. So using kind of a consensus-based approach. So this is also commonly done in pathology, where not this specific thing, but it's common to kind of use the wisdom of the crowds. So what we're doing here is to validate the accuracy of the models, or to know that we're as good as the model is possible to get. We randomly sample sub-image frames across a data set. These are data set samples that are not part of the training set. Ask 5 pathologists or 7 pathologists or some panel of pathologists to label each and every single nucleus within that panel, and then compare our models with that of the pathologist. So that's kind of our frames-based validation approach. We kind of shared this at a SITC presentation a couple years back. We have kind of elaborated on this method quite a bit. We try to take into consideration each and every pathologist's biases when doing this type of analysis. We think it's absolutely critical and important to know that when we're predicting different cell types that later we want to analyze the features derived from those predictions, we want to make sure that they're as accurate as possible. More
Great.
questions?
Another question that just came in: can these models be applied to multiple tumor types?
Yeah, so we typically have tumor-specific models. So we have like a lung cancer model, we have a breast cancer model, gastric cancer model, I think this is addressing the question: can it be applied to similar tumor types? We almost always would have a starting place when we're looking at, let's say it's a new cancer type like endometrial or cervical, something that we don't necessarily have a lot of experience with. We would probably start with a model that has as much similarity in terms of tissue or cellular morphology and composition to that kind of target data set. Then we inevitably have to go through some period of model refinement and corrections to make sure that it's capturing those features as possible. Another kind of analog to this, like, so you could take the same tumor, like maybe you have a model that's trained very, very well on like primary tumor sites, and now you want to deploy it on a data set that is metastatic. So we would probably start with that tumor site and then refine the model to make sure that it is able to capture how the disease presents in the kind of the target data set. And then once we were happy with the performance, again, with pathologists that kind of review the model and frames validation to see the performance of the cell predictions, finally, we would then validate it on kind of a standalone validation data set to really understand how it's going to perform on data that hasn't really been part of the training process.
Great. Another one here. What is the typical turnaround time for accessing these algorithms? And maybe more broadly, Ilan, you can touch on how folks can work with PathAI as we think about sort of the 3 diamonds that you showed previously.
Yeah, so a lot of the models that we have are pretty automated, right? So that the goal here is to be as automated as possible. But we also understand that each and every data set has its own peculiarities. Sometimes there's the format, sometimes it's we want to make sure that the model deployment is kind of fit for purpose and we give the right attention that it deserves. So deploying the model is quite instantaneous, right? It takes just minutes to kind of to hit go on the button. There's a series of models that get deployed. I didn't talk about our artifact detector model or QC models, but there's kind of layers of models that go into each of these deployments, and they happen very, very quickly. We could do on thousands of slides within days. Typically, a turnaround time, just kind of the high level question, it's 1 to 2 weeks is what we try to aim for. And that is kind of the end to end. So onboarding the data set, QCing the data set, deploying the models, QCing the results, having discussions with our partner, and making sure that they understand how best to interpret the features, making sure that the features themselves are QC'd, et cetera. So high level, 1 to 2 weeks is kind of like the short answer for that long answer.
And sort of related to that, do we have to provide whole slide images? So are there other ways to work with PathAI, say if there are not whole slide images of H&E readily available?
Yeah, no. So for model training, we have access to data sets that are, I mean, unless you have a very niche or rare disease, let's say, that it's quite easy for PathAI now to partner with our data partners, such as Cleveland Clinic. We have now access to quite a few venues. We have also a lab in Memphis that has its own patient samples that we're able to, in many instances, access for model training. And we offer that to partners. So that's something that we could do. There's always some kind of, there's carve outs for what is possible, what isn't possible. But that's in terms of model training. In terms of the deployment, again, there it depends on the question, but oftentimes our partners want to deploy our models and get features on their data sets. So we would need their data sets we need them to upload feature slides to our platform. We deploy our algorithms, and then we return the features.
It's something that we didn't spend very much time today speaking to, but as Ilan mentioned, PathAI is also operating a laboratory. And so in cases where whole slide images are not available, we can work with everything from tissue as well as glass as a starting point as we think about the process through scanning and digitization.
That's a really important point. And I'll just mention one thing about that, because the quality of the digital image is actually quite important in determining some of the quality of the resulting analyses and how well the models can perform and interpret those features. So one thing that has been very beneficial for PathAI and for the partners that have used this is the ability to kind of have those data sets scanned through our facility, and then we deploy our models and return the features, and we also return the digitized images for other purposes.
Okay, it looks like we're coming up on time, but I think this is a really great question to end on. Are there any plans for clinical diagnostics, or perhaps more broadly, Ilan, if you even want to speak to potential applications that you see for tumor and microenvironment and the models that you described beyond translational research?
Yeah, so for instance, I won't go into too many details, but yes, there's beyond translational research, a lot of these models can be used for clinical development. I think I touched on some of that, or I think this is a big goal for PathAI. We can use these algorithms for instance, patient screening and selection, for identifying which patients are good candidates to have downstream molecular tests drawn from them, or for enrolling into different clinical trials. So it's kind of on that level, just for enabling efficiencies for the clinical development process. And then downstream of that, for diagnostics, et cetera, I think there's, again, a very good use case here where a lot of the basic diagnoses that this technology enables can be standardized, made more reproducible and more accurate once that's kind of implemented and developed for clinical use. There's kind of a long ways to go before we get to some of those applications, but we always keep an eye towards that, and that is where the technology is heading. That's where the field is heading. That's where PathAI wants to go.
Great, thank you. And thank you all for joining. We apologize if we weren't able to get to your question, but please feel free to reach out. You can see Ilan's email here, and we're happy to continue discussing both this topic and any other questions that may arise over the course of the conversation. And so with that, I'd like to thank you all for joining, and we'll see you all next time.
Thanks, everyone. Thanks, Ryan.